Hi everyone, welcome back to Bobo's Biology channel.
I am Mark.
And I am Celine.
And we are the Bobo's Biology team.
In this episode, we'll explain the basics of neurobiology.
From action potentials to the different parts of the neuron. We will discuss it here. Neurobiology is the study of the brain, spinal cord, and nerves. It's all about how cells communicate, process information, and control everything we do, from breathing to thinking.
Or in Bobo's case, daydreaming about cheese. So let's start our journey into the depths of the brain.
Welcome aboard the microscopic flight from the lab to the brain. Please enjoy your flight.
Oh, do you see this? What we have here is a multipolar neuron.
Oh yes, that's the most common type of axon in the central nervous system. It is composed of one axon and multiple dendrites. First up, neurons. They're the brain's messengers.
So as we can see, the dendrites receive the input from other neurons via neurotransmitters.
There are different types of neurotransmitters. Keep this in mind when we discuss them later. Now we are at the cell body, also called the soma,
which is equipped with everything a eukaryotic cell has, like a nucleus, the Golgi apparatus, mitochondria, ribosomes, and endoplasmic reticulum. But keep in mind that neurons have a higher mitochondrial density than most other cell types.
That's the case since neurons constantly need to maintain an iron gradient across the membranes, which requires ATP.
Mitochondria, therefore, tend to cluster in synapses and nodes of Ranvier, which are small unmyelinated gaps between the myelin sheath segment along the axon.
And fun fact, the myelin sheaths you were talking about are made from roughly 70 to 80% of lipids and 20 to 30% of proteins. Their function is to be an excellent electrical insulator that lets the action potential skip over the parts of the axon covered by myelin, which makes the action potential travel much faster than in unmyelinated axons.
But now, back to our neuron. There goes the action potential depolarizing the axon hillock, which is located in the spike triggering zone. It is called this way since it sets off the new action potential along the axon, which we'll come to shortly.
The axon hillock is composed of the highest density of voltage-gated sodium channels. If the signal received from the previous neuron is strong enough, they help trigger an onset of the action potential down our axon.
To do this, the axon hillock receives excitatory and inhibitory postsynaptic potentials called EPSP and IPSP from the dendrites, and then integrates them into one.
Exactly, and if these postsynaptic potentials combined at the axon hillock reach a certain threshold, then a new action potential is created. At rest, the neuron's membrane potential, so the electrical potential difference between the inside and outside of the neuron, sits at about -70 millivolts. The sodium-potassium pump quietly pushes out 3 positive sodium ions and pumps in 2 molecules of positive potassium ions, therefore keeping the concentration gradient across the membrane of the neuron. So the pump pushes out more positive charge than it brings in. It pushes out 3 positive charges, but brings in only 2. This alone makes the inside slightly more negative relative to the outside of the neuron.
The sodium-potassium pump uses ATP to maintain the gradient. Additionally, at resting potential, the potassium leak channels allow for positive potassium ions to leak out. Since the inside concentration gradient of potassium is higher than the outside, the potassium ion passively diffuses out through the leak channels.
Further, the sodium leak channels, which are less abundant than the potassium leak channels, also allow a few ions to diffuse with their concentration gradient across the membrane. So the intracellular fluid concentration of potassium is therefore higher than the extracellular, and the concentration of sodium is intracellular lower in relation to the extracellular concentration at resting potential.
If you think now that it kind of seems like the sodium-potassium pump and the leak channels are working against each other, you're not wrong to think that at first. It sure sounds like it, but like in every time in biology, it has a very good reason to exist in the first place.
If there were only leak channels, the gradients would eventually even out. Sodium and potassium would equalize and the neuron could never fire. If there were only the sodium-potassium pump with no leak channels, the membrane potential wouldn't stabilize at minus 70 millivolts. The ions would be locked in imbalance and continue to build up, but wouldn't set up a usable electrical state. Think of it as a charged battery, just waiting for the signal. When the threshold is reached at minus 55 millivolts, voltage-gated sodium channels snap open. Voltage-gated channels are transmembrane proteins that open or close in response to changes in electrical potential across the cell membrane, allowing specific ions like sodium or potassium to flow through the membrane. The channels are ion-specific, so there is response specifically for sodium or potassium, but not for both.
When the voltage-gated channels open, sodium ions flood into the neuron because they are attracted to the inside since it's more negative than the outside. And chemically speaking, the concentration gradient is pushing them inside because the concentration of sodium ions on the inside is lower. That sudden rush makes the inside of the cell less negative to about positive 30 to 40 millivolts. Keep in mind, the positive 30 to 40 millivolts is only for the inside of the cell. It's not the membrane potential. The membrane potential during depolarization is negative 55 millivolts.
Now, the neuron doesn't stay excited forever. Almost immediately, sodium channels close and voltage-gated potassium channels swing open.
Potassium ions rush out, bringing the membrane potential back down to minus 70 millivolts. That's repolarization, the neuron restoring its negative inside.
But potassium channels are a little slow to close, so the neuron dips below the resting potential to about minus 75 to minus 80 millivolts for a short time.
That's called hyperpolarization. The neuron becomes extra negative, which makes it harder for another action potential to start immediately. This is the refractory period.
Here's the coolest part. The action potential doesn't just happen in one spot. It travels down the axon.
In unmyelinated axons, it moves step by step, each section of the membrane depolarizing the next. In myelinated axon, it jumps between nodes of Ranvier in a leapfrog style called saltatory conduction. Way faster.
So, to summarize, sodium goes in, potassium goes out, the charge is reset, and the signal races down the axon to the synapse.
All of this happens in just a few milliseconds. Neurons are like the fastest messengers in biology.
Alright, we followed the action potential zooming down the axon, but what happens when it reaches the end?
That's the synapse, the tiny gap between neurons. And here's where neurotransmitters do their magic.
When the action potential reaches the axon terminal, it triggers voltage-gated calcium channels to open, which are found primarily at the axon terminals of the neurons.
Exactly. Calcium ions rush in, and that's the signal for synaptic vesicles to move toward the membrane and release neurotransmitters into the synaptic cleft.
Neurotransmitters are like tiny chemical messengers. Once released, they diffuse across the synaptic cleft, which is only about 20 to 40 nanometers wide.
And then they bind to specific receptors on the next neuron's postsynaptic membrane, kind of like a key fitting into a lock. These are called liganded gated channels and open only in response to a chemical messenger, for example, a neurotransmitter.
Once a neurotransmitter has bound, it induces a conformational change in the channel, which opens the pore of the channel. This can, depending on the neurotransmitter that bound to the channel and the type of channel itself, have two different outcomes. For example, glutamate, the most common excitatory neurotransmitter in the brain, binds to AMPA or NMDA receptors.
When glutamate binds, these channels open and sodium ions flow into the neuron. Because sodium is positively charged, this makes the inside less negative, a process called depolarization. This creates an excitatory postsynaptic potential, or EPSP. If enough EPSPs add up at the axon hillock, the neuron reaches threshold and fires a new action potential.
On the flip side, GABA is a common inhibitory neurotransmitter. It binds to GABAA receptors, which are also ligand-gated.
When GABA binds, these channels let chloride ions flow in. Since chloride is negative, the inside becomes even more negative, hyperpolarized, making it harder for the neuron to fire.
This produces an inhibitory postsynaptic potential, or IPSP. Basically, GABA is putting the brakes on the neuron. So every neuron is constantly summing up EPSPs and IPSPs from all its inputs.
If the net effect reaches threshold at the axon hillock, a new action potential fires, and the signal continues. If not, the neuron stays quiet. But before we move on to the world-renowned Bobo's Brainbusters, let's catch our breath with some fun facts about neurobiology.
So for today's fun facts, we have hands, hair, and a runner. What could possibly be the connection between those?
Neurobiology, of course. Fun fact number 1. Did you know that neurons never touch? You heard that right. The synaptic cleft is about 20 to 40 nanometers wide. That's about 1/2500th of a human hair.
Fun fact number 2. Your brain's extremely fast. Some action potentials can travel as fast as 120 meters per second. That's way faster than an Olympic sprinter.
Now let's test your knowledge with Bobo's Brainbusters.
Question number 1. What is the resting membrane potential of a typical neuron?
Option A, plus 40 millivolts. Option B, minus 70 millivolts. Option C, 0 millivolts. Option D, minus 50 millivolts. And the correct answer is option B, minus 70 millivolts. Question 2. Where are the voltage-gated calcium channels primarily located?
Is it A, in the dendrites, B, in the soma, C, in the axon terminals, or D, in the nodes of Ranvier? And the correct answer is C, in the axon terminals. Question number 3. Which neurotransmitter usually inhibits postsynaptic neurons?
Option A, glutamate. Option B, GABA. Option C, acetylcholine. Option D, dopamine. And the correct answer is option B, GABA. Question 4. What happens when sodium rushes into a neuron?
Is it A, hyperpolarization, B, depolarization, C, the neuron shrinks, or D, the axon myelinates? And the correct answer is B, depolarization. That was it with today's video about neurobiology basics. We discussed the action potential and the different parts of the neuron, as well as different neurotransmitters.
We hope you liked it. Please let us know how you like our new style of videos, and if you have any suggestions, we really appreciate your feedback. Please don't forget to like and subscribe to our channel. See you in the next video and stay curious.